what I'll do now is I'll give a quick overview of what this workshop is really about, why we've started it. So I joined Imperial's bioengineering department in 2016, and when I did, the first person I met really was Guy, and Guy is in synthetic biology, and my background is in nucleic acid nanotechnology, and Guy was very excited about the idea of using nucleic acid nanotechnology in synthetic biology, and I was very much saying, "Oh yes, nucleic acid nanotechnology is great. You can design incredible things. For example, these dynamical systems from Srinivas et al. A few years ago. You can basically design arbitrary reactions to go at arbitrary rates, and you can do whatever you like." And Guy was very enthused by that possibility of including that into synthetic biology, which more traditionally works on the transcription factor circuitry level. And there are several reasons why he was interested in that. One in particular was the idea that building transcription factor circuits is quite burdensome for the cell, and it might be better if we could do things at a nucleic acid level, and other things to do with predictability and programmability of the circuits that you're building. Now, although there have been a number of impressive works published taking steps in this direction, we're obviously still quite a long way from implementing the sort of circuits such as from Eric's group shown on the right here in living cells, or even systems that are sort of artificial approximations of living cells. And one question we had was, well, what was stopping us? And in the specific context of dynamic DNA nanotechnology, what I kind of realized at this time was that the way we build nucleic acid systems to take part in dynamic DNA nanotechnology is typically based on strand displacement technology, and that works as shown in the top of this figure here. And typically speaking, the way strand displacement works is that we chemically synthesize a series of oligonucleotides. Here there's 3 separate oligonucleotides chemically synthesized. We anneal 2 of them together in a pot, and then we separately add a third one, and that third one triggers a displacement reaction, and we've got one single reaction where we release an output in response to an input. And that's for one reaction. If we were to implement a big network of reactions, such as in Eric's group work on the previous slide, we'd have lots of separate syntheses and lots of separate pots where we mix the strands together. And what we realized was it's actually quite difficult to do this in an autonomous system such as a cell in situ. It's quite difficult to get a cell to produce these basic components, and in particular these gates, where you have 2 or more strands in a complex that are very important in determining what series of reactions occur in response to an input. And one reason it's difficult is, I mean, you might say, well, cells have got the machinery that we need. They've got RNA polymerase. We can make arbitrary strands of RNA in a cell. So why can't we just make the components of this circuit? And the reason that doesn't work, well, one reason is these 2 strands here in this complex that you make, the strand displacement gate, they need to be made in an appropriate stoichiometric ratio, and it's very difficult to guarantee that if you're getting the ingredients transcribed on the go. But arguably even more important is that they're not produced in the same place. If you produce 2 separate strands of RNA and then hope that they come together and form the strand displacement gate before you reach before they interact with any of the other strands in the system, that's a recipe for failure. It's basically you can't use the pre-preparedness of these metastable strand displacement gates. If you're transcribing all the strands separately, they might interact with each other out of order, and that essentially all the logic of dynamic DNA nanotechnology breaks down. So we submitted a grant to try and get around this problem, and our idea was to build multi-stranded gates via transcription, and our idea was to transcribe a single strand that folded into a hairpin and then chopped itself up using self-excising ribonucleases and self-nicking strands basically, and produced complexes on the fly that could be used as these strand displacement gates. And I won't go into that in any more detail, but that is the topic of Woolly's poster, which is poster number 13. So we proposed that, and we said, okay, let's try and build that, and we have indeed been able to build something like that. At the same time, we thought it's worth thinking, if we really can build these RNA circuits and put them in cells, what kind of theoretical framework should we be thinking of to enable these nucleic acid circuits to function in a more heterogeneous and challenging environment than perhaps ideally prepared in vitro test tubes? And here we had Tomislav working on the project, and he was thinking a lot about controller networks. So here the idea is that you have some reaction going on, some process going on, but because it's subject to external fluctuations, it's useful to have another circuit which is controlling various aspects of your original circuit to prevent them getting out of control. And he's been thinking a lot about how to do that, and he's presenting some of it in poster 21. Now, we submitted this work and it was funded, but as part of that proposal, we promised to organize a workshop. And what we were saying is, we're going to bring nucleic acid nanotechnology to synthetic biology. And in order for that to be a success, what we need is the nanotechnologists to get together with the synthetic biologists and say, yep, this is great, we can really use these ideas and push it forward. And so that's why we started to organize the workshop, which originally was going to be in person, but of course now is remote. However, the project actually got us thinking a bit more deeply about the similarities and differences between the sort of things that occur in nature and get used in synthetic biology and the sort of things that we create typically in nucleic acid nanotechnology. And so actually, in the end, the workshop has grown in scope, I guess you could say, from just this purely, let's try and get nucleic acid nanotechnologists and synthetic biologists to do stuff together, to a more sort of like philosophical perspective. So the question, one of the main questions we're trying to address in this workshop now is, can or should nucleic acid nanotechnology be more lifelike? So in this example controller system I showed here that Tomislav was thinking about, the way it works is that you have a circuit you're trying to control, and you have your controller circuit, and the controller circuit is continuously operating. It's continuously, maybe indefinitely for all time, working to keep the input circuit within the bounds of whatever it's aiming to achieve. Similarly, if we look at an actual natural system, for example, chemotaxis in E. coli, chemotaxis in E. coli is always ongoing. It's always passing on a signal about whether to turn the motor in one direction or another in order to respond to a continuously changing signal of the concentration of food in the outside world. This is a bit different from how we usually design nucleic acid nanotechnology circuits. So this example from Lulu Chan's group, beautiful set of beautiful system designed to perform pattern recognition, essentially using just nucleic acid strands. It's very good at it, but it works once. You put in the strands that symbolize an L or a T, and you get your answer. But then if you want to do it again, you need a completely new system. So this is a very different sort of paradigm where things fire once or things continuously operate. One other aspect in which nucleic acid nanotechnology tends to be different from how living systems work is that living systems tend to create far from equilibrium states, whereas quite often nucleic acid systems, synthetic ones, we're building things that relax to some stable equilibrium. So although the non-equilibrium nature of the process is important to begin with, the thing we end up with is often equilibrium. So a good example, I mean, life is doing this all the time, creating non-equilibrium states. This F0 F1 ATP synthase is converting electrochemical gradients into a free energy, high free energy molecule, creating a non-equilibrium distribution of ATP. That would be just one example. Here's our conventional strand displacement reaction, and typically what happens is that we start off with this non-equilibrium configuration where we have an input duplex and an input single strand, and the single strand displaces the output and reaches a more stable state, which is an equilibrium state. Now, this also applies to self-assembly, structural nucleic acid nanotechnology. So these are images from Peng Yin's lab, the incredible versatility and functionality of his DNA brick system for self-assembling structures. This is an amazing achievement, but fundamentally what's happened here is his group of cleverly designed a set of strands to find a very stable equilibrium that is that the system is able to reach. By contrast, possibly the most important assembly operations that happen in living cells are the assembly of biopolymers, DNA, RNA, and proteins. And those processes, as you will find out if you go and chat to Javier and Jordi, my students, are fundamentally operating out of equilibrium. The aim is to produce a non-equilibrium product rather than an equilibrium product. So really the physics, the underlying chemistry is very different. And related to this, living systems often rely more on catalysis, genuine catalysis, where we have a molecule that comes in, interacts with something, changes it, comes out, and is retained. Nucleic acid nanotechnology, there are catalytic systems, but it's less prominent than in natural systems. And the example I've shown here is the kinase cascade, where signal transmission is done via catalytic activation of subsequent steps in the cascade, rather than signals being consumed as they typically are in strand displacement. Not always, but typically. And in fact, the processes I talked about on the previous slide, these production of biopolymers like RNA, DNA, and so on, these are also using a catalyst. They're using the template strand that encodes for the product is acting as a catalyst. And we don't really have in nucleic acid nanotechnology things that do catalytic templating of assembly at the moment. So we do things differently. Does it matter? Would it help to do things more like natural systems? Are there some things ideas we should take and others we should ignore? That's kind of the topic of this workshop. So more of our group members will be presenting work in this direction. I've already mentioned Javi and Jordi, who have got some very exciting experimental and theoretical results on templating of non-equilibrium systems. So basically trying to build synthetic analogs of transcription and translation. And Francesca will also be presenting work on modeling strand displacement more accurately. And really, this is very important because when you start building these non-equilibrium systems, you have much less freedom to play with free energies. and you really have to be able to tune what you're doing very precisely. Okay, so I've said that our groups are working on these questions, but obviously we're not the only people who are working on these questions. And typically when I've said DNA nanotechnology is like this, I'm making a very general statement, and there are lots of people who are trying to push it in a different direction. And I'm not saying that that work hasn't been done. And in fact, we've invited these people to speak because they've influenced us across a range of topics. So based on the presentations that we're going to see, we're specifically hoping to generate discussion around 3 questions. The first is, what do we need to do to make DNA or RNA nanotech in living cells more predictable and usable? The second is, in what ways should we be looking to further emulate natural molecular systems? Should we be trying to do things differently from natural systems? Is there a fundamental advantage we can have to doing things differently? And thirdly, what key applications or features can DNA RNA achieve in living cells? So Guy will be chairing a discussion on these points on the final day. We've created a channel, it's at the bottom of the Teams page, where you can post ideas and we will return to them in the discussion. Okay, yep, that's it. So these questions, they're in the conference, the workshop handbook, and they're also on the general page. But yeah, if you can try to think about them, that would be absolutely fantastic. So, Guy, what time are we on?
We are bang on time, 1:30.
Okay, brilliant. Well, thank you for listening to me. With all of that preamble out of the way, it is time to get stuck into the good stuff. So let me stop sharing my screen.